The discovery of the genetic code was a revolutionary milestone in science, which allowed information encoded in RNA sequences to be converted into polypeptide chains of functional proteins. Numerous studies contributed to cracking the genetic code by establishing its nature. For example, a study by Francis Crick and Sydney Brenner in 1961 was influential in clarifying codons were read in a non-overlapping nature. However, in 1957, Brenner was remarkably able to deduce the non-overlapping nature of codons from a simple statistical analysis in his paper on the impossibility of all overlapping triplet codes in information transfer from nucleic acid to proteins. At the time of Brenner's study, it was known each of the 20 amino acids was determined by a combination of 3 nucleotides, and that therefore there were 64 codon possibilities, making the genetic code degenerate. It was also believed codons were read in an overlapping nature, whereby a dipeptide would be encoded by 4 nucleotides. It was theorized overlapping would protect against frameshift mutations and help condense the genome, as roughly 1 nucleotide would correspond to 1 amino acid. Brenner realized there were 256 possible dipeptide nucleotide sequences, and hence dipeptides, in an overlapping system. However, there were actually 400 possible dipeptide combinations, meaning with overlapping, some dipeptides could not be made. Brenner sought to solve this problem by finding more than 256 dipeptides to refute overlapping. However, at the time, fewer than 256 dipeptides were found, as only few fragmented amino acid sequences were mapped. Instead, Brenner used those mapped sequences as a sample of the data to statistically show overlapping was not possible. Brenner deduced that if overlapping occurred, the C and N neighbor codons would each share 2 nucleotides with the central codon, meaning they were restricted to being only 1 of 4 codons from the total 64 codons. Knowing this, Brenner analyzed the few mapped amino acid fragments, studying each amino acid and seeing which amino acids could be observed as its neighbors. For example, for histidine, he observed 6 amino acids could be its C neighbor and 9 amino acids could be its N neighbor. Taking the larger number, Brenner was able to calculate the minimum number of central codons required to code histidine. For example, if CAU was a codon for histidine, possible N neighbor codons would be UCA, ACA, CCA, and GCA, which could possibly code for 4 different amino acids that were observed to be histidine's N neighbor. Another codon, for example UGC, could code for histidine, resulting in 4 more N neighbor codons, each which could code for another 4 different amino acids observed as histidine's N neighbor. This still leaves 1 amino acid that was observed as histidine's N neighbor unaccounted for. Therefore, 3 codons are needed to code for histidine in order to accommodate all its N neighbors in an overlapping system. Using this logic, Brenner calculated the minimum required number of codons for each amino acid and realized a total 70 codons would be needed. However, at the time, it was known only 64 codon combinations were used in the genetic code, hence overlapping could not be possible. Therefore, like counting passengers on a train multiple times, overlapping nucleotide readings would result in an overestimation of the actual number of codons, or carriages, needed. It was previously thought with a 1-to-1 ratio of amino acids to nucleotides, protein synthesis would occur with amino acids lining up along the RNA strand and an enzyme moving along to link them. However, the implications of Brenner's results meant non-overlapping, and hence a 1-to-3 ratio, which suggested the polypeptide growing at the contact point of the nucleotide sequence, something later discovered to be true. His results also meant all 400 dipeptides were possible, increasing the variability of proteins. Brenner's simplistic original study was remarkably able to statistically deduce the non-overlapping nature of the genetic code. His study also provided the foundation for his future research with Crick, whereby they worked with frameshift mutations to experimentally determine the non-overlapping nature of the genetic code. The contribution Brenner and many other scientists made in cracking the genetic code revolutionized the field of genetics, allowing scientists and students alike to be able to simply decode RNA sequences into amino acid polypeptide chains.